morning. Thank you for joining me this afternoon. My name is Matt Levy. About 2 years ago, I was a full-time employee of Carestream Health, and for the last 2 years, I have managed an in-vivo imaging core facility at a university in northern Indiana. So every once in a while, I like to take off my academic hat and put back on my Carestream hat and discuss some of their technology and applications with customers. And so today we're going to talk about the specific application of imaging cell death mediated by apoptosis or necrosis in living models. So first I want to tell you a little bit about the product offering from Carestream, and it's a unique one, 2 instruments that can supply 7 imaging modalities. So here at the top you will see the multispectral, which has, of course, multispectral fluorescence and luminescence, as well as X-ray and radioisotopic imaging modalities. A new product that was launched just a couple months ago is the Albira tri-modal system, which offers 3-dimensional PET, positron emission tomography, 3D SPECT, or single photon emission computed tomography, or CT, which is just X-ray computed tomography. So each of these instruments has their strengths, and certainly with the multispectral FX, you have a platform with which to perform rapid, multimodal, planar imaging. In this case, you get these 4 modalities that I mentioned before: multispectral fluorescence, luminescence, radio imaging, and X-ray. And I think that this model you could perform with this instrument. You can collect a variety of signals, whether they're multispectral fluorescence, luminescent, or radioisotopic, and overlay those on an X-ray to perform something called multi-modal imaging, where you use the X-ray as an anatomical map to look at the anatomical origin of your various signals. And here you can see we have a variety of signals emanating from the heart, kidneys, GI tract, thyroid, brain, lungs, and liver, and of course we have many other examples. So while this is a rapid and convenient planar imaging platform, Carestream now offers a 3D platform through the use of the Albira tri-modal system. And of course the modalities offered here, as I mentioned before, are PET, SPECT, and CT, and I've shown a couple classic examples down here below where now you're again using the X-ray anatomical scaffold that you're utilizing, and you're then superimposing your molecular signals emanated from radio tracers in the PET or SPECT modality. So for the rest of the talk, I'm going to focus on strategies for imaging cell death in vivo. And the way you typically do that is identify a couple of biochemical pathways or biomarkers that take place or get upregulated or activated during the cell death process. So the 3 that we're going to look at here are, number 1, imaging of metabolic changes. So one can imagine that metabolism would be reduced in cells and tissue that are dying. We'll look at the activation of Caspases and probes that can be used for that, as well as probes for the detection of phosphatidylserine, which is apoptosis. So it's hidden until a cell undergoes programmed cell death, and then it becomes exposed. And it's also prevalent on the cells of necrotic tissue as well, because the membrane is permeabilized, and thus your probes can leak in to detect phosphatidylserine in that fashion. So first, let's look at imaging with FDG. So what is FDG? Well, it's a very simple probe in that it is a radio-labeled sugar. So it's F18 labeled D-glucose. And as one can imagine, this is a nice analog of glucose that is readily absorbed in tissue as if it's the real sugar. And of course, it's going to be preferentially taken up in cells with high metabolic activity. And the beautiful thing about this probe is that when it's internalized, it's then phosphorylated, thus trapping the FDG within the cell. So things like the heart and brain, which of course are using a lot of sugar in their native state, but also things like tumors, which are undergoing cell division and thus require a lot of energy, as well as inflammatory lesions. So I'm going to show a couple examples of using FDG to perform three-dimensional imaging on the Albira trimodal system. So here you see a PET-CT fusion image of F18 FDG accumulated in a tumor-bearing mouse. So as I mentioned before, you see the X-ray, which is providing the three-dimensional scaffold, and you see areas of hotspots or localization of the FDG, and those include the kidney as well as the bladder. And here we have a tumor that's at the base of the tail that's also taking up the probe as well, and thus highlighting its location. So another example from this system, another early example, is the use of FDG to monitor therapeutic efficacy. So in this case, 2 HeLa tumors were going up on the legs of immune mice. And then each of the tumors was treated with a therapeutic dose of radiation, either 25 grays or 10 grays. Afterwards, mice were imaged using the Albira PET-CT system, and we wanted to see is there a correlation between radiotherapy dose and imaging. So here's the animal in the CT modality, and this shows you each tumor that was irradiated. And when you use traditional measurements like using a caliper through the skin or using a CT to look at tumor volume, we saw no noticeable change in the size of these tumors after irradiation. But when we went and looked as noted, or as judged by F18 uptake, we saw that in both cases the tumors had, relatively little uptake and retention, probably because they both had a radiation dose. But in this case, the top tumor did have the least uptake, and that correlates with the fact that it received the highest radiation dose. And so this is a nice example that shows proof of principle that you can indeed use FDG to look at and correlate changes in metabolism with radiation dose. So, whereas now let's talk about radio imaging, I'm going to move and speak to examples that deal with optical imaging. Specifically, we're going to be looking at fluorescent probes that can be used during whole animal fluorescence imaging. So optical techniques are emerging as a very popular strategy with which to do preclinical studies because of the ease of use of the probes. So first, let's talk about Caspase activatable probes. Caspases are themselves activated during the process of cell death. So one strategy would be to comprise an activatable probe that would be cleaved by a caspase and thus result in an increase in the fluorescence from the probe. So researchers in the lab of Dave's Pinnacle Worms have recently accomplished this task by using a peptide with the classic DEVD recognition sequence that's recognized by Caspase 3 and 7. And they use this DEVD sequence to connect an electrophore, a near-infrared electrophore, with a quencher. Now, this isn't enough for just a probe because you still have to get into cells, and thus they've added a tap peptide on the end of this to facilitate cell uptake. You will have a cleavage at this DEVD site. The electrophore will dissociate away and thus can give it a full breadth of its fluorescence without worry of the quencher. And so they tested their probe in vivo using human colonic xenografts and then infected those xenografts with amoebas to cause apoptosis. And here are a couple examples using what they call the TCAP-Q, so tap peptide, caspase, and quencher is what that means. And here are 2 mice that were injected in this study, and you can see uptake in this tumor and activation of the probe over time. Now, when we use a DTCAP-Q, so that means that the scrambled recognition sequence, there was no activation noted, and of course in the standard control there was also nothing. So these fluorescence uptake values correlate with the tunnel score gathered during histology, and you can see with increasing amounts of fluorescent signal, there were increasing amounts of apoptosis noted. Now, they've gone on to use this TCAP2 probe in a number of different models, and perhaps this is one of the advantages of using fluorescence is that it's amenable to other fluorescence techniques, for example, microscopy or flow cytometry. So here they've gone on to use their fluorescent probe in a model of glaucoma, where they're injecting NMDA into the retina, inducing cell damage, and here you can see three concentrations, 5, 25, and 80 micromolar NMDA, and then subsequently injecting the probe and then extracting the eyes for purposes of microscopy. And there's no doubt that you can see a matter apoptotic and thus Caspase, and thus have Caspase activation after an NMDA treatment. So here is another example of a probe. This is from the Lattice Stomatilosu, where now we're trying to use multimodal imaging to look at the presence of the probe as well as Caspase activation. So the core of the probe is the same with your recognition sequence, fluorophore, and quencher, but at the end, rather than having a tap peptide, they've now included accumulation moieties for radio tracer. So what they did to test the feasibility of this compound is they performed two experiments. First, they injected the unquenched peptide through the animal, or through an animal, and saw that it lights up and gives a significant amount of fluorophore. Whereas the quenched peptide, which is this animal. Next, they performed a phantom study in which they took capsules and took one capsule and included Caspase-3 within the capsule, and another one is without Caspase-3. And after they added the Caspase as well as, added the Caspase to the capsule, they subsequently added their probe. And what you can see here in the fluorescence modality is that indeed there's a great deal of activation and thus increased fluorescent signal coming from the capsule that was included with Caspase versus the one that did not. However, in the radioisotopic imaging modality, or doing basic scintigraphy, you can see that there was relatively almost identical amounts of signal being emanated, and that's what you'd expect because there was no activation of the radioisotopic signal in this case. So here you're using multimodal imaging to look at the location and distribution of the probe, and then also look to see if the probe is being activated. So finally, I'm going to talk about probes for the detection of phosphatidylserine, which I mentioned is exposed on cells that are undergoing apoptosis, as well as detected during cells that have died by necrosis due to membrane permeability. So here is the structure of Annexin-5, and this is a classic compound that's used for the detection of phosphatidylserine, which it binds in a calcium-dependent manner. Now, the work of myself and my collaborators, the work of me and my collaborators, has focused on producing small synthetic mimics of Annexin-5 that can achieve the same goal. And we've done that through using these dipicolylamine zinc coordination complexes and attaching those to fluorophores. And here I've shown anthracene, a thing for phosphatidylserine sensor with a molecular weight of around 1,000 versus 40,000 for the Annexin. Now, as an initial test, we would perform microscopy to verify that our compounds, our PSS-based compounds, are functioning in similar fashion to Annexin. So here is a study looking at apoptotic Jurkat cells. And what you can see is that we have a populations of cells which are circled, which essentially stain blue or green, but do not stain red. And this red, which is called 7-AAD, is a DNA stain which marks cells that have been had membrane permeabilization. And as I mentioned before, these cells still stain blue, but we know that the membrane permeabilized and thus not apoptotic by looking at 7-AAD. So, as one might expect, nice co-registration and co-localization with a FITC labeled Annexin-5 compound. So next, in order to facilitate in vivo imaging studies, we've taken out the anthracene and incorporated a near-infrared Di7 similar fluorescent. And the reason we've got it in near-infrared is because these photons exhibit the best tissue penetration. So we took this compound, and first we looked at it in a series of tumor imaging studies. And the reason we've done this is that some tumors, as they grow, the inside of the tumor becomes sort of, if you will, strangled off from nutrients in the bloodstream, and thus becomes necrotic. So the necrotic core of the tumor would be a target of our In-Vivo probe, and indeed you can see that it was published this year if you'd like to go in and take a quick look. Now, as part of the same studies, we also looked at prostate tumors that are growing in the flanks of rats, and when we took out the tumor and dissected it, we could see that most of the signal was coming from within the inside of the tumor, which caused us to hypothesize that indeed we are finding the necrotic core and dead tissue within the tumor. I'll also add that PSF794 is now commercially available from a company called MTTI, which stands for Molecular Targeting Technologies Incorporated. So, histological analysis of the tumor I showed you in the previous slide showed that indeed we are staining dead cells as compared to live cells. And so in this case, the tumor was, after its ex vivo imaging, it was frozen in OCT, and we took sections of that and imaged them using near-infrared microscopy to figure out the probe localization. In the phase contrast field, you can see cells that are clearly deformed and thus reason to be dead, whereas this is a normal-looking tissue, and we see very little uptake there. So moving on to other models, we want to show and demonstrate and validate the PSF794 compound and its ability to stain dead tissue in a highly controlled animal model of dead tissue. And so what we've done in this case to reduce any biological variability that might occur, say, in stroke models or even in tumors or heart disease models, is to initiate a very rapid tissue damage response from 3 different chemical agents. And we use ethanol, and we use injectable anesthetic that just happens to cause a little bit of tissue damage, as well as a synthetic channel. So here I'm showing a cohort of 4 mice that was injected in its right leg with a synthetic ion channel that disrupts ion homeostasis and thus causes cell death. So here are all 4 mice from the cohort, and you can see over time that the probe definitely binds, especially within 3 hours you get a very nice localization, and over 24 hours the probe goes to the rest of the animal, and we see that we're getting very nice and bright staining from these. And so when you visualize the whole cohort, what you see right away is that indeed this is a robust model for testing the PSF794 compound, in that all the animals are virtually identical, and that the 794 compound is indeed doing its hypothesized duty in binding this tissue. And here at bottom I've taken the final image, fresh a little bit, and overlaid it on X-ray for anatomical probe registration. Now, as we look at 3 different animals, here we are showing a typical animal from the hydrophile cohort, and the ethanol cohort, and the ketamine cohort. In each case, we see localized tissue damage to this muscle tissue when we use PSF794. When we use an untargeted control probe, we get no localization here. We just see it as it's clearing through the intestine. So, one thing I'd like to show during these types of talks is how do you perform quantitative measurements when you're using an optical modality like fluorescence? And the answer is that you use ratiometric imaging. And what that means is you will calculate such things as target to non-target ratio. So here we'll get full intensity of the target region divided by the non-target. So this is a ratiometric way to elucidate the relative amount of probe that was involved in each of these sites. And what you can see is whenever we use 2, this is the TSS794 compound, we get very good target to non-target ratios in each of the 3 models, ranging from 6 to 4 to around 3, whereas with compound 3, which is the untargeted control, and so that means we're simply taking the, let me go back one more, we're taking this fluorescent part and removing the affinity group. When we just look at the 4-4 alone, then we get very low uptake, and it's basically at or near 1. Of course, we've also done histology here, and what we see from the periphery of these same cell bodies. And finally, what we wanted to do is perform a direct comparison of TSS794 to an annexin conjugate that had a similar fluorophore with similar spectral properties. And indeed, we were able to use Annexin-Vivo 750, which is available from Bison Medical. So at left, you see a typical montage from our tissue damage studies, looking at an animal pre-injection and then at 3, 6, 12, and 24 hours post, and we can see the uptake of our probe. When we try to use the annexin, however, based on the clearance pathways through the bladder, we're basically unable to perform measurements at the 3, 6, and 12 hour time points. However, by 24 hours, we have enough clearance that we can visually see that we've also performed a 48-hour image, which has even further clearance of the dye, and indeed, we see the same thing. So the annexin results certainly verify that phosphorylserine was exposed in this tissue model, but our take-home message and our conclusion from this is that perhaps our probe is more appropriate for this type of this type of tissue damage model, where you're looking at the legs in a sort of highly controlled system, versus over here where you're looking at the legs and the bladder is saturating at your signal. Now, perhaps TSS794 is a good complement, because if we were looking at tissue damage in the liver, we would suffer the same consequences, since the liver is the clearance pathway for TSS794, and the bladder is the pathway for Annexin-Vivo 750. So our conclusion today from this presentation probes target different pathways. Further, PET-CT imaging reveals changes in metabolic activity in both tumors and tissues, and there are other nice probes available that are activatable, that could report on the presence of activated caspases or on the availability of phosphorylserine within anionic membrane surfaces. Finally, multimodal imaging certainly provides researchers the tools to look at multiple pathways in cell death and evaluate potential therapeutics in your preclinical small animal model. So with that, I'll say thanks, and if you'd like more information, feel free to contact CareStream Health or visit the webpage, or shoot me an email at matthew.levy@gmail.com. Thank you for your time, and have a good day.